Okay, so welcome back to this next video in which we are discussing the receptor tyrosine kinases. Okay, right, so we're in the process of discussing the different families of receptor tyrosine kinases. Now, we're not going to go through them all because there are actually 20 different families. We're just going to look at 7 of them, the 7 most famous. Okay, so we've seen the B family, which remember contains the receptors for epidermal growth factor. We've seen the PDGF family, which contains receptors for platelet-derived growth factor. We've seen the VEGF family, which contains receptors for vascular endothelial growth factor. And finally, we've seen the FGF family, which contains members which are receptive to fibroblast growth factor. Okay, the next family of receptor tyrosine kinases we're going to talk about is the TRK family, which is pronounced TRK. Okay, so we're going to talk about the TRK family of receptor tyrosine kinases. Okay, so we'll start off once again with their structure, and then we'll talk about examples of TRK family receptor tyrosine kinases and what their respective ligands are. Okay, so once again, let's let this be the phospholipid bilayer here. We then have the N-terminus of our polypeptide extracellularly, and we then have 3 domains making up the ligand binding domain here. Okay, so 2 of these are going to be immunoglobulin-like domains, once again, and one of them is going to be a leucine-rich region. Okay, so we'll colour in the leucine-rich region in green here. Okay, then we'll colour in the 2 immunoglobulin-like domains in blue here. Okay, so those 2 are immunoglobulin-like domains, 1 and 2 here. So I'll just put IG for immunoglobulin and then like domains. Okay, and then we've also got this region which is rich in leucine amino acids. Okay, so this is the leucine-rich region. Right. Okay, then after the second immunoglobulin-like domain that we've got here, we then have the membrane-spanning alpha helix, and then we have our tyrosine kinase domain, and in the case of the TRK receptors, the tyrosine kinase domain is whole and intact. Okay, so it's one large stretch of the primary structure that makes up the tyrosine kinase domain here in turquoise. Okay, so this is our tyrosine kinase domain. Right, so now let's discuss the examples of receptor tyrosine kinases which fall into this 5th family that I'm talking about of receptor tyrosine kinases. Okay, so there are 3 main receptors in this family. The TRK-A receptors, the TRK-B receptor, TRK-B, and then finally the TRK-C receptor as well. Okay, right. Now they all have different ligands. Okay, so let's talk about the different ligands for the different TRK receptors. So the ligand firstly then for TRK-A. What is the ligand for TRK-A? The ligand for TRK-A is something called nerve growth factor, so another growth factor. Okay, so NGF is short for nerve growth factor. Okay, and its receptor is the TRK-A receptor. TRK-B, what's the ligand for TRK-B? TRK-B has a ligand known as BDNF, okay? And this stands for brain-derived nerve factor, or rather, brain-derived neurotrophic factor. Okay, so the B is for brain, the D is for derived, and then the N is for neurotrophic, and then the F is for factor. Okay, so this is also a factor involved in the growth of neurons. Okay, and its receptor is TRK-B. Then finally, for TRK-C, another factor involved in the growth of neurons, known as NT3. Okay, now NT3 stands for neurotrophin-3. So the N, once again, is for neuro, the T is for trophin, and then the 3 is for free. Okay, so TRK-C receptors, their ligand is NT3 neurotrophin-3. Right, finally moving on to my 6th family, okay, which completes this simple sort of receptor tyrosine kinase And the final family after that I want to talk about is the insulin receptor family, but they're quite different, okay? So we'll discuss those separately. So the next family is what's known as the Efamily, which is a very large family of receptor tyrosine kinases. This has a lot of receptors within it. Okay, right. So once again, we'll draw the phospholipid bilayer here. Okay, then we'll have our amino terminus here. Okay, now all Efamily receptor tyrosine kinases have a special domain near their amino terminus known as an ephrin binding domain. Okay, because the ligands for Eph receptors are going to be ephrins. Okay, so this is a special domain for binding to ephrins. So I'll colour the ephrin binding domain in green here. Then after the ephrin binding domain, what you then have is two domains which you find in many different proteins, okay? So you have two of a certain type of domain, two copies of this certain type of domain, okay? And it's known as a type 3 fibronectin domain, okay? So both of these are type 3 fibronectin domains. So we have two of these type 3 fibronectin domains after the ephrin binding domain here. Okay, so just like immunoglobulin-like domains, type 3 fibronectin domains are domains that you find in many different types of proteins. And when we do discuss the insulin receptor, we'll see that there are also these type 3 fibronectin domains within insulin receptor subunits as well. Okay, right. Then you'll have the membrane-spanning alpha helix again. So I'll colour that in red here. Okay, and once again, the tyrosine kinase domain here is going to be whole and complete. So the region that makes up the tyrosine kinase domain is going to be one continuous region within the primary structure of the polypeptide for these ephrin family receptor tyrosine kinases. Okay, and then you have the C-terminus intracellularly. So that's the structure of these Efamily receptor tyrosine kinases. So what examples of receptor tyrosine kinases come under this family of Efamily? Okay, so there are 9 receptors which are all coupled together as being EphA receptors. Okay, and these are labelled up EphA1 all the way along till EphA8. Okay, so it goes EphA1, EphA2, EphA3, EphA4, all the way up to EphA8. Then there is no EphA9, and then it goes straight on to EphA10 to give us overall 9 EphA receptors. Okay, then as far as the EphB receptors are concerned, you have 5 of these, and you have EphB1, EphB2, EphB3, EphB4, then there is no EphB5, and then we go straight on to EphB6. Okay, so those then are our Efamily receptor tyrosine kinases here in red. Okay, right, so that concludes my discussion of these first 6 families of receptor tyrosine kinases. As I say, there are many others. There are another, it goes up, there are 20 total families of receptor tyrosine kinases. We're going to see 7 of these, okay? The 7th one that I haven't shown you yet is the insulin family, which is slightly different from all the others. The other 19 are all monomeric, like so, and they all have the same mechanism of activation, basically. So what I would now like to discuss is this mechanism of activation of all of these 6 families of receptor tyrosine kinases that I've now discussed. Then we'll come on to the insulin receptor. We'll see how it's different from the other receptor tyrosine kinases and how its mechanism of activation, therefore, is also different. Okay, right, so let's now discuss how these receptor tyrosine kinases are going to activate. So, hopefully you'll agree that we can pretty much abbreviate all of these different families of receptor tyrosine kinases that we've discussed now to just having an amino terminus extracellularly, then some sort of ligand binding domain. Okay, whatever this is. So we'll just colour this in orange. This is the domain which is going to bind to the ligand. Okay, so we'll call this the ligand binding domain, or the LBD for short. Then after the ligand binding domain, we have our membrane spanning alpha helix. And then on the cytoplasmic domain, we either have a split tyrosine kinase or a whole tyrosine kinase. It doesn't really make that much difference. The point is we've got a tyrosine kinase enzyme down here. Okay, so here is our tyrosine kinase enzyme, or tyrosine kinase domain rather, which is going to phosphorylate tyrosine residues within proteins. And then we have our C-terminal. Okay, right, so these things wander around in the cell membrane, basically, until their ligand arrives. So we now want to have a look at how they're going to be activated. So let's now say the ligand has bound to the ligand binding domain of our receptor tyrosine kinase here, of our generic receptor tyrosine kinase. And we're going to keep this completely general. We're not going to name any names here. So we're just going to say this is a ligand, basically, that is now bound to our ligand binding domain. Okay, so here's our tyrosine kinase domain, and here's our C-terminal tail. So let's colour everything in. So here in orange, this once again is our ligand binding domain. I think I'll put a colour for the membrane spanning region as well. There's our membrane spanning alpha helix. Okay, and here is our tyrosine kinase domain intracellularly. Okay, then we have our ligand in blue here, bound to our ligand binding domain. So this blue box here, this is our ligand. Okay, now what will happen once the ligand binds to the ligand binding domain like this? Well, it will trigger a conformational change in the receptor that will now mean that it can dimerize with another one of these identical receptor tyrosine kinases, which also has a ligand bound. So here comes along our other identical receptor tyrosine kinase here. Here's the amino terminus, okay? Here is our membrane spanning alpha helix here, okay? Here is our tyrosine kinase domain, and here is our C-terminal tail here. Okay, and I shouldn't have drawn the two tyrosine kinases joined together like that. That's misleading. They don't join together. Okay, so it's the extracellular portion that binds together like this. So here are the two tyrosine kinase domains separated. Okay, right, but the ligand binding domains have now bound together. Ooh, and that second receptor tyrosine kinase should have a ligand bound to it as well. Now, for our purposes, we will assume that these two receptor tyrosine kinases are utterly identical. Okay, so we will assume that this is a receptor tyrosine kinase and this is an identical protein. Okay, when this is true, then what this is called is a homodimer. Okay, so homo means the same, dimer means two-membered structure. So this is a structure with two identical receptor tyrosine kinases. Okay, so it's a receptor tyrosine kinase homodimer, if you like. Okay, now, what can happen is sometimes receptor tyrosine kinases can dimerize with other receptor tyrosine kinases which have ligands bound to them, which are not identical to them. Okay, so for instance, let's say this is a receptor tyrosine kinase of the ERB1 type. Okay, so let's say this is an ERB1 receptor tyrosine kinase, which is, remember, the other name for the epidermal growth factor receptor, or HER1. Okay, right. So that makes this ligand the epidermal growth factor. So what can happen is it can either bind to another identical epidermal growth factor receptor with epidermal growth factor bound to it and form an epidermal growth factor receptor homodimer, or it could bind to another receptor tyrosine kinase, providing that this other receptor tyrosine kinase is in the same family of receptor tyrosine kinases as it. Okay, so it can bind to another ERB receptor tyrosine kinase, so for instance an ERB2, okay, a HER2 potentially. So if this HER2 receptor tyrosine kinase or ERB2 receptor tyrosine kinase had an EGF molecule also bound, then those two receptor tyrosine kinases with their ligands could dimerize together, and that would be what's known as a heterodimer. Okay, so you can form heterodimers between two different receptor tyrosine kinases, but in order for this to occur, they have to be within the same family. So you can't have two ridiculously different receptor tyrosine kinases binding together, because remember, they need to have the same sort of ligand binding domain here, and we talked about the fact that receptor tyrosine kinases that aren't in the same family don't have similar ligand binding domains, whilst those in the same family do. Okay, right, so what then follows this dimerization is that these tyrosine kinase domains that we have here are now going to take part in a process known as Transautophosphorylation, or merely just as autophosphorylation. So I'll write down this massive great word in a moment. Okay, after I've finished labeling up the two tyrosine kinase domains. So they're going to participate in a process, the full name for which is Transautophosphorylation. Okay, however, people will often just call this autophosphorylation. So often this is just called autophosphorylation. Okay, so what's now going to happen then in autophosphorylation? So basically these tyrosine kinase domains, usually in the receptor tyrosine kinase that has no ligand bound, this tyrosine kinase domain has a very low level of vasoactivity. Okay, so it can phosphorylate tyrosine residues, but it does it incredibly slowly, basically. So what's now going to happen is when you bring the two receptor tyrosine kinases together like this, you're going to get this tyrosine kinase domain here. Okay, we'll call this tyrosine kinase domain number 1, and we'll call this one here tyrosine kinase domain number 2. Tyrosine kinase domain number 1 is going to phosphorylate tyrosine residues within tyrosine kinase domain number 2. So remember, this is a protein, it has tyrosines within it. Some of these are going to get phosphorylated, at least one of them is going to get phosphorylated by the tyrosine kinase domain on receptor 1 here. Okay, so it's going to put phosphate groups onto tyrosine residues in tyrosine kinase domain number 2. And then tyrosine kinase domain number 2 will also reciprocate for tyrosine kinase domain number 1. So basically, both of these tyrosine kinase domains are going to phosphorylate residues within the opposite tyrosine kinase domain. Okay, now what does this result in? Well, the addition of phosphate groups onto the tyrosine residues within these tyrosine kinase domain causes activation of the tyrosine kinase domains. So these two enzymes become much more active once they've now been phosphorylated by the opposite one. Okay, now this process is known as transauthophosphorylation, okay, or merely just as autophosphorylation. Auto means self. Phosphorylation means adding phosphate groups onto yourself, onto something. So autophosphorylation means adding phosphate groups onto yourself. Now, this is why transauthophosphorylation is a slightly better name, because the reason it's called autophosphorylation is you view the dimer as being one structure now, and therefore the dimer is phosphorylating itself. These tyrosine residues are getting phosphorylated by the dimer's own domains, basically. Okay, so that's why it's called self-phosphorylation. But then the transauthophosphorylation kind of recognizes the fact that actually this is a dimer of two separate receptor tyrosine kinases, and that it's one phosphorylating the other rather than one phosphorylating itself. Okay, so the autophosphorylation kind of takes into account that the dimer is now being viewed as one structure. The trans takes into account that, it's two separate proteins, basically. Okay, right. So, then what happens is once these tyrosine kinase domains have been activated, they're then going to go wild, basically. They're then going to phosphorylate tyrosine residues all over the place in the cytoplasmic tail of the other. So, okay, let's draw this picture out again and show the next stage. So I might draw a bigger picture now so that I've got more space to play with. Okay, so here is our membrane spanning alpha helix. here are two ligand binding domains which are dimerized together. Okay, then we've got our now activated tyrosine kinase domains here. Okay, and to show that they're now activated, I will stick little blobs off the side which will represent the phosphate groups which they have had added onto them. Okay, so let's put a little bit of colour on here. So here we have our tyrosine kinase domains here in turquoise. Okay, and they have been phosphorylated in this transautophosphorylation process, and therefore have these phosphate groups now on them, at least one. Okay, then we have our membrane-spanning alpha helices here in red. We have our ligand binding domains which have dimerized together, and I'll show these in orange here. Okay, like so. Then we have our amino termini extracellularly up here, and we have ligand bound to these ligand binding domains extracellularly here. Okay, and we'll colour this in blue. One ligand molecule here, and another ligand molecule here. Okay, right. So these tyrosine kinase domains, they're now activated, and what's going to happen is the tyrosine kinase domain here on the tyrosine kinase receptor, or the receptor tyrosine kinase rather, number 1, is going to phosphorylate all sorts of tyrosine residues on the cytoplasmic tail of receptor tyrosine kinase number 2. Okay, so we're going to put phosphate groups onto tyrosine residues all over the place, basically. One here, one here. Okay, meanwhile, the activated tyrosine kinase domain on receptor tyrosine kinase number 2 is going to do the same thing for receptor tyrosine kinase number 1. So we're going to get phosphate groups put onto the tyrosine residues on the cytoplasmic tail. Okay, right, so we're going to create many of these phosphotyrosine residues. Okay, right, and what's now going to happen is that onto these phosphotyrosine residues, proteins are going to dock. Okay, and we'll look at examples of the signaling pathways downstream of these later. Okay, but for now, what's happened is we've got loads of these phosphotyrosine residues on the intracellular side of our receptor. Okay, what can happen is that proteins can come and bind to these phosphotyrosine residues. So there are important domains that proteins can have which are capable of binding to phosphotyrosine residues. I'll discuss these two special domains now. So let's say this is representing a protein. Basically, in order to bind to a phosphotyrosine residue, let's say this is a tyrosine tyrosine residue with a phosphate group coming off. Okay, you need to have there are special conserved domains within proteins which are capable of binding to phosphotyrosine residues. And two examples of these, the main two examples, are the SH2 domain, okay, which is a conserved structure that you find in many different proteins, and the name stands for Sark homology 2. Okay, so S for Sark, H for homology, and then 2 for 2. So the Sark homology 2 domain, okay, and this is a very common domain that you find in all sorts of proteins that interact with receptor tyrosine kinases, okay, which is capable of binding to tyrosine residues with phosphate groups on, i.e. phosphotyrosine residues. And by the way, the single letter amino acid code for tyrosine is Y, which is why I've put a Y there. Okay, so this domain, you find it in many different proteins, and if a protein has that domain, often it will be able to bind to phosphotyrosine residues on the cytoplasmic tails of these activated receptor tyrosine kinases. Okay, now, another domain that's less common that also has a similar function to the SH2 domain is the phosphotyrosine binding domain. Okay, which I think is called the PTD. In fact, it might be called it is called the PTD, I think. It's called the PTB. That's what I'm thinking of. Phosphotyrosine, yes, that's it. Not the PTD. Right, so PTB domains, okay? So PT obviously stands for phosphotyrosine, and then the B then stands for binding. So phosphotyrosine binding domains, or a PTB domain. Okay, so these are less common than SH2 domains. SH2 domains, you find them in all sorts of proteins, but in some proteins you instead find these phosphotyrosine binding domains. Okay, and these, the principle is the same, that they can bind to phosphotyrosine residues within proteins. Okay, so when you have one of these activated receptor tyrosine kinase dimers like so, proteins with phosphotyrosine binding domains will be able to come and dock onto these phosphotyrosine residues. Okay, now, in the case of some proteins, just binding to the phosphotyrosine residue will activate them. Okay, so the protein may well be activated just by the binding of the phosphotyrosine residue. In addition, sometimes it's not the case that binding to the phosphotyrosine residue will activate you, but instead, just by binding to this phosphotyrosine residue, it doesn't change your activity, but it locates you at the membrane. Okay, so maybe you had a protein that was in the cytoplasm, and in the cytoplasm it was active, but it didn't have its substrate. Maybe its substrate is in the membrane. Okay, so it wasn't actually capable of doing anything in the cytoplasm, even though it was active. Now, when the receptor tyrosine kinases activate, it will now bind to these phosphotyrosine residues here, and maybe the binding won't affect its activity, but now that it's bound there, it's in the right place to gain access to its substrate in the phospholipid bilayer, and it can then perform its function. Okay, so sometimes just binding to the phosphotyrosine residues within the cytoplasmic tails of these receptor tyrosine kinase dimers just serves to relocate the enzyme to the right place, basically. Okay. In addition, finally, some proteins will come and bind to these phosphotyrosine residues on the intracellular aspect of these receptor tyrosine kinase dimers, and the binding of to the phosphotyrosine residue won't activate the protein. But what will happen is it will latch onto one of these, and then it's close in proximity to the tyrosine kinase domain of the receptor tyrosine kinase. Okay, so if I draw this here, let's say this is our phosphotyrosine residue here, bound to either an SH2 domain or a PTB domain here. Okay, but then now what's happened is we've positioned our protein extremely close to this tyrosine kinase domain. Okay, binding to the phosphotyrosine residue hasn't actually activated the protein, but it's positioned it beautifully close to this tyrosine kinase domain here. And now what will happen is the tyrosine kinase domain will phosphorylate a tyrosine residue on this protein, and that will then change the function of this protein. Okay, so sometimes there are proteins whose function will be changed by the tyrosine kinase. So the phosphotyrosine residue docks them close to the tyrosine kinase domain, which we know is now activated because it's got this phosphate group that's resulted from autophosphorylation. Okay, right. So that's the activation then of these most receptor tyrosine kinases. Okay, what we'll turn our attention to in the next video is the insulin receptor. We'll look at its structure, and we'll also look at how its structure means that its activation is going to have to be slightly different.